Hi everybody, welcome to another session of cell biology. So in this session, session 4, we're going to look at cell communication signaling, and we're also going to look at the cell cycle. So, if you remember from last session, the types of communication, there were two types of communication. So transport or trafficking, which was to do with vesicular transport, and also signaling. We did the transport trafficking last time. This time we're going to talk about the signaling. Okay, so a bit of revision. What are the different types of vesicular transport? So if you remember, there was 3 types of vesicular transport. There was endocytosis, of which there was 3 types. Okay, there was receptor-mediated, phagocytosis, and bulk phase endocytosis, otherwise known as pinocytosis. There was exocytosis and there was transcytosis. So for trafficking, vesicular transport, it's the movement of vesicles in and out of the cell membrane, or out of the cell, through the cell membrane. So now we are going to do signaling, which was transport of a specific signal received from the outside to elicit a cellular response, be that inside the cell. So, definition. Signaling is linked to transport and trafficking, so it's where specific molecules secreted from cells by exocytosis act as signaling molecules. So what that means is molecules or substances are taken out or sent out of the cell, and those particular substances then act as signaling molecules for another cell. So there's 3 types of signaling. You have neural signaling, paracrine signaling, and endocrine signaling. So in neural signaling, you can see here you have a neuron. It's called a presynaptic. You'll learn more about that when we do nervous system. But you can see you have a presynaptic neuron, which is actually a cell. So it's a particular type of cell. And what happens there, that cell releases proteins called neurotransmitters, which are these little structures here that you can see, and they go and attach to the postsynaptic neuron, so other neuron cells. And they act downstream on a target cell. So it may be a muscle cell or it may be another neuron. So that's neural signaling. Another form of signaling is paracrine signaling. So you have a signaling cell, so you've got this particular cell here that and it secretes protein molecules. These are the little protein molecules here. Okay, so they're secreted from the cell and they act locally on target cells. So instead of going downstream, they just act locally. Okay, and it ensures functioning of cells in very close proximity. So this is what happens when we're talking about tissues, which we'll talk in the next module. And then the last type of signaling you've got is endocrine signaling. So this is where you've got an endocrine cell, such as a cell in the thyroid, something like that. It releases a hormone, so you can see the hormone here. It then goes into the circulation, which is the bloodstream, and then it gets presented to a target cell that is further on down the track. Okay, so the 3 different types of signaling: neural signaling, paracrine signaling, and endocrine signaling. So when we're talking about the signaling molecule itself, okay, you have a signaling cell. Now please note, when I did these diagrams, you've got the diagram has got a single L. That's very American spelling, and in Australia we spell it with 2 L's. It really doesn't matter, but just so you know, it's the same thing. So we've got a signaling molecule that is released from the signaling cell. So in this example here, you can see the little signaling molecule are these 3 little red dots. So it can be a hormone, it can be a neurotransmitter, or it can be any other protein secreted from or by a cell. That signaling molecule then goes and attaches to a receptor on a target cell. Okay, so the target cell and the receptor will depend on the signaling molecule. So this green receptor here can only accept this little red signaling molecule because it's the only one that fits into the lock and key. So an example there is acetylcholine, which is a neurotransmitter. It's secreted from a motor neuron cell. So this would be a neuron here, for example. It would be a neuron cell. and then acetylcholine will be released and it will go and attach to the receptor on a muscle. And as long as that muscle contains the acetylcholine receptor. So what it means is there's specificity of response. So only a target cell with the right receptor will respond to the signaling molecule. We've got a bit of revision here. So when we're talking about receptors, is receptor an integral or a peripheral protein? Okay, so you need to go back and have a look at that. Think about that question and you can answer it. If you can't answer it straight off, go back and have a look. So when we're talking about how signaling works, we've got the signaling molecule here that's already been released from the signaling cell and it goes and attaches to a receptor. So what it is, it's the process of converting an extracellular signal, and that signal is the signaling molecule. So it's process of converting a signal, which is extracellular because it comes in from the exterior, into a intracellular response. So that may be, it may alter the metabolism of the cell, it may result in gene expression, or it could change the cell shape or motility. And it does that via a transduction cascade. Now, all of that, so cell signaling, that process is initiated by the extracellular signal and receptor engagement. So you can see here, this is the cell, so this rectangle, black box, is the cell. Around it is the cell membrane, and in that cell membrane you have a receptor. And that extracellular signal has come and attached to the receptor. And what that sets into play is something called a transduction cascade. And you can see these are these purple boxes here. Okay? And then what it ultimately ends up in is an intracellular response, which are these red responses here. So the transduction cascade, okay, is initiated by, as we've said before, the signal receptor engagement, which is this area here, and it leads to amplification of the signal. So what happens in this transduction cascade, so this is the purple section here, okay, first of all, it activates something called a second messenger system. So this is this box here, so it attaches, so the signal attaches, signal is a molecule, be it a neurotransmitter or a hormone, and it goes and attaches to the receptor, and then that sets in play the transduction cascade. So first off, you have a secondary messenger system. It could be a G protein or it could be cyclic AMP. Then that activates protein phosphorylation cascade, so that's the next step. And then an example there is AKT. Don't worry too much about the particular names such as AKT, just understand the concept of what happens. And then what that does is that transduction cascade then activates specific target proteins within the cell. Okay? So when it talks about amplification, it's saying that one signal and one receptor can ultimately lead to multiple responses. So you can see one situation up here results in 3 situations down here. So that's what they're talking about when they talk about amplification. So we've got here, we've gone from the extracellular signal attaching to the receptor, which brings into play the transduction cascade. And what that does then, it activates target proteins. Now those possible target proteins within the cell could be transcription factors. We've already talked about transcription somewhere. If you remember, it's to do with central dogma, okay? And it's all about gene expression, okay? That's what the central dogma is. It's about those genes expressing themselves and creating a protein. It also, a target protein can be an enzyme, and we've talked about enzymes before. A particular example of one was RNA polymerase, or lactase, or anything ending in ase is an enzyme. And another target protein that it could activate are cytoskeletal or skeletal proteins. So if you remember the particular proteins that were involved in the cytoskeleton, you had intermediate filaments, you had microfilaments, and you had microtubules. Now, as a result of activation of those target proteins, which is here in this blue section, that then results in the response. And those responses might be gene expression, which is production of a protein, so that's where your central dogma comes in. It could be metabolism alteration, so it could change some reaction in the cell. Or it could be involved in cell motility and shape alteration. Okay, so they're the three responses that it could lead to. Now, just a bit of revision. What are the two processes involved in gene expression? So when we're talking about gene expression, we're talking about the central dogma. So have a think about what are those two processes involved in that, in the correct order. When you always talk about them, make sure you talk about them in the correct order. So we're talking about transcription and translation. If you can't remember the three processes involved in those, the three steps involved in those processes, make sure you go back and do a bit of revision. There's also a video down here that you can look at, the YouTube video, and it is talking about signaling with gene expression as a cellular response. Now, the signaling molecules work in various combinations to regulate the behavior of a cell. So ultimately, a cell has four things that it will do. So the overall response. Now, the first one is to survive. So it doesn't divide, it doesn't do anything, it just lives. The next one is it can grow and divide. So that is cell division. So one of its responses, with all of this transduction cascade and signaling, it might signal the cell to divide. Another thing that it may make the cell do, so the signaling may make the cell do, is to differentiate. So what that is a progenitor cell differentiates into a mature cell. So it starts as an immature cell, and the signaling cascade says, okay, now you need to differentiate into a different type of cell. And then the last overall response is death. So it might get told to die. There are two types of death. There is apoptosis, okay, new word for you, which is pre-programmed cell death. So every cell has a length of time it will survive for, and then after that the cell tells it to die. For example, a red blood cell has a lifespan of 120 days. After 120 days, that cell should die. If the cell doesn't die when it's told to, that can mean cancer. Now, another way that it can die is necrosis, and that's where it's damaged in some form before it was due to die. So just a little thing down here. Can you think of a cellular process that would be able to control the cell's destiny? Okay, and another one is name situations where each of these cell destinies are important for maintaining health. So I've talked a little bit about that, so just have another big think about that. Very simplified, so the previous slides that we looked at was a very simplified way of saying what happens in a cell as far as cell signaling is concerned. But actually in a cell, this is what it might look like, how it really looks. There is so much going on with one signal, giving messages to all different things in the cell to ultimately come up with a response, which would be cytokine gene transcription. Okay, so all of these messages are going on. So that's actually what cell signaling is about. Come to the end of this one, so you can now go to session 4 of your tutorial and look at activity 1 and complete activity 1 about cell signaling. Okay, we'll have a break and I'll see you in the next one. Bye.